Hi, this is my AP Capstone project on genetically modified human embryos. As you can see, designer babies. So what exactly is a designer baby? Well, according to the Oxford English Dictionary, it is a baby whose genetic makeup has been selected in order to eradicate a particular defect or ensure that a particular gene is present. This can be anything from, as the picture shows, a high IQ, getting rid of inherited diseases, athletic skills, perfect vision, just basically, a designer baby. So the first subtopic I want to discuss is in vitro fertilization, also known as IVF. And that's how the idea of genetically modified human embryos came to be. It's how basically they got the idea off of. IVF is the process of fertilization via extracting eggs, retrieving a sperm sample, followed by manually combining an egg and sperm in a laboratory dish. As you can see, this is pretty much what it looks like, where they basically inject a sperm cell into an egg. Of course, it doesn't actually look like this in real life, but it just gives you like a clear visualization of what happens. So the link between IVF and designer babies. So genetically modified human embryos are originated from embryos created via IVF and are selected either because of the presence or absence of a particular gene, or because an embryo was created by genetic interventions in order to attempt to influence the traits of the resulting fetus will have. This can be anything from diseases, like if they have a higher chance of inheriting diseases such as the BRCA gene, or gender, basically like any characteristic. Okay, CRISPR. This is how it's possible for genomes of human embryos to be altered. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a mouthful. So CRISPR, in short, in the very simplest version, is a genome engineering technology that enables scientists to easily and precisely edit the DNA of any genome. So as you can see in this picture, this, of course, it doesn't actually look like this, it just is for visualization, where, they're cutting a part of the DNA to either get rid of a gene or just alter the genome as they please, pretty much. So all about CRISPR. So CRISPR works by having the Cas9 enzyme edit genes by precisely cutting the DNA and then letting natural DNA repair processes to take over, like the picture in the previous slide. Geneticists can alter the genomes of IVF embryos using this. So the application of CRISPR. So the work, despite the ethics, still being, discussed, it's actually being done right now in China. An example of this is a team at the Southern University of Science and Technology in Shenzhen, excuse my pronunciation, is working to eliminate the gene CCR5 using CRISPR in hopes that the offspring will be resistant to HIV, smallpox, and cholera. As you can see on the right here, this is a magazine cover of when people realized that this was being done, and that basically the trial was successful. One giant step for designer babies. So the third subtopic is the ethics. Genome editing presents many difficult challenges and debates regarding ethics. Sorry about if you can hear my dog in the background. We just got a new puppy and he's very feisty. So a quote that I liked, that I found regarding the ethics is, the concern is that with technologies that are relatively easy to use, like CRISPR, how does the scientific community regulate itself? This is by Robert Truog, again, excuse my pronunciation, and he's the director of the Center for Bioethics at Harvard Med School. And I completely agree with him. Like once you find out, because it really is easy to use. Like what stops them from basically building a apex human? So ethics can also differ based on the situation. So somatic engineering versus germline engineering is one example. So in somatic, it affects only the patient being treated and only some of his or her cells. And then germline affects all cells in an organism, including the eggs and sperm, and thus is passed on to future generations. And then another one is IVF versus genome editing. So with IVF, it's the embryonic development that can be halted by the action of a physician with the likely consent of the couple. The question here is, was the embryo wronged? Is it a human life? Does it have rights? And this is still, very controversial among certain groups. And then genome editing. The thing with genome editing is that possible consequences are hard to predict. And a lot of people ask if humans are wrong to play Mother Nature or even God when it comes to the genomes of all future generations. This map is basically the legal landscape of where everything, like the levels and the restrictions, are placed. So as you can see in places like China, it's permissive but regulated, versus Mexico, it's permissive by legislation. And then in places like Brazil and Australia and Canada, it's restrictive by legislation. And then you can see the source of where I got this at the bottom. Thank you so much for watching this, and this was such a great class. And these are all my references. Thank you.